notes. Have you started with the perspective based notes? No, not yet. You have to start with the perspective based notes. Post level 1, it is perspective based notes. October 31st is the deadline for you to complete perspective based notes. When you are doing perspective based notes, you have to just run through the textbook to find out what you want. Like, tell me the perspectives. Recall the perspectives. Okay, we'll do with biology. What are all the perspectives on which you will make notes? Glossary, or the terminologies and their one sentence description of what it is. It's like a dictionary. Glossary is a dictionary of your textbook. All right. Second, then diagrams, tables, glossary, diagrams, tables. If there are any flow charts, then mind map, you already have made it. Syllabus map is the mind map. Then anything else? For chemistry, what all do you do? Glossary is common for everything. Glossary, tables, diagrams are common for everything. All subjects you have to look at the subject in these three perspectives: glossary, tables and diagrams. Then in chemistry and physics you will have formulas, you will have derivations, laws. If there are any laws, you have to make note of it. The laws of physics, laws of chemistry, all those things have to be made note of. You will not read the entire chapter, you will just look for those. For making a glossary, when you just run through the textbook, you can take a pencil and underline the terms that you want to include in the glossary. Just underline those words. Add those words and write one sentence description of that. That's all. For diagrams, you know, as you keep going through the textbook, you will find out the diagrams. Try to make the diagrams in simple line diagram format. Simple line diagrams, neatly labeled. While labeling, you should make sure that, let's say, if this is the diagram, labeling should be organized. All right, it should not be like this, like this, like this. No. A neatly organized labeled diagram will help you revise faster. The entire job till October 31st is to build revision material, basic revision material, and beyond that it is to build continuously developing revision material. All right. Your performance is dependent on what you do in the last 1 month. April will decide how you perform on May 4th. But how you do in April is determined by what you do through the rest of the year. All right, what you are doing right now is to ensure that you have well organized, well developed, comprehensive revision material by April, which you have already revised multiple times. All right. So you need to know, performance is not just about how much you have prepared or how long you have prepared or how many years you have prepared. Nothing of that matters. You have to peak at the right time. If you peak at the right time, if you are in the best preparation mindset on the day of exam, that is the best exam, that is the best performance for you. And that is determined by 1 month before that. That 1 month before the exam is determined by 6 to 8 months before that. That is what you are doing. So stick to the process, completely work on building the revision tools. We will also give all of that prepared by us. All right. So by the time I leave this weekend, I will share the syllabus map of both 11th and 12th standard. Okay. I'll open it now and tomorrow's class I will take it in the evening, 5:30 to 8:30, 9:30 to 12:30. I have to go to Shillong to meet the governor. Governor is from Karnataka. I have taken an appointment. I just have to go and meet and greet him. Why is it? There is no internet. Who can fix this? Oh, it's turned off. Huh? Every time you turn it off?
No, sir.
Okay. Reload. Open a new tab. If it shows Gmail automatically. Yes. Okay. Open the first one. Download it. You all have got your textbooks, no? One set of textbooks for everybody. And today you will get the performance tracker booklet. Performance tracker booklet. Whatever I want you to note down it can be noted down in one booklet. So one booklet for you to be preserved forever. During this year it will tell you how you are progressing. You can keep it forever to show people later. Alright. You have to build all of that story. You have to show that. 20 years later, it is very valuable. I could get access to something that I had written for our college magazine about myself about 18 years ago, 17, 18 years ago. It feels good when you look back what you are thinking and what you have done. Where is it? You downloaded it? Did you download it? We had done about 8 chapters, isn't it? Visibility at the back close? No? Better? No? Turn off these lights. These lights. No? Better? Plant kingdom we discussed, animal kingdom we saw. Can you recall the 10 phyla in the animal kingdom? Tell the list of 10 phylum in the animal kingdom. Okay. Anybody else? Okay, collectively tell. First one is Porifera. Porifera. Coelenterata, pseudocoelom. Then Ctenophora or Cnidaria. Then Platyhelminthes flatworms, Ashkelminthes roundworms. Then Annelids, Annelida, jointed organisms, arthropods, insects, jointed legs. Then mollusca, soft body, external shell. Then Echinodermata, spiny skin, spiny body. Then hemichordata, chordata. All right. 5 divisions of plant kingdom. 5 divisions of plant kingdom. What are the 5 divisions of plant kingdom? First one is fungus-like body, undifferentiated body, which is Thallophyta. Thallophyta, then Bryophyta, Pteridophyta, Gymnospermae, Angiospermae. Okay. See, these things should be on your fingertips. That is where the syllabus map will help you. Get the anchors first. You need to get the anchors. The anchors will help you expand. Alright? Okay. Till what did we complete? Structural organization in animals did we complete? No, we did till the plant anatomy. Till flowering plant anatomy we did, no? No? Okay. Structural organization in animals. You all have your tabs with you right now? No? Would it be better if I share the document with you and then all of us see in our respective devices and do it? Okay. Turn on Bluetooth, one of you. Not working? What happened? Oh, you dropped it, no?
No.
It's not switching on. We get it repaired. Turn on Bluetooth. One sentence description wherever required also has been given. I have given one sentence description of, simple description of what each is. Simple epithelium, single layer of cells, covers body surfaces. See, when I say body surfaces, it is not only external. Wherever there is an surface, it is covered by epithelium. So the inside of the blood vessels is also a surface to which something can come in contact. So that is also covered by epithelium. What is the epithelium of the inside of the blood vessel called? It has a different name. Endothelium. The difference between epithelium in other places and endothelium is that the endothelium is super smooth. The surface is very smooth to reduce friction for the flow of blood. Compound epithelium is multiple layer cells, provides protection. Squamous epithelium, thin flat cells. Wherever you need exchange as the priority, wherever you have exchange as the priority, you will have squamous epithelium. Wherever you need stability and structural strength, you will have columnar epithelium. So a balance of both will lead you to different types of epithelium. Where you need some stability but movement is more important, cuboidal epithelium. It provides better stability and strength than squamous epithelium, but a little lesser in terms of exchangeability, the ability to allow for exchange. Columnar epithelium, you look at it in your tab, in your document, I'll see here. Columnar epithelium will give you more strength, more stability, but it will compromise on the exchangeability, exchange of materials. So wherever you want, wherever the priority is strength and not necessarily exchange, you will have columnar epithelium. Where you need more exchange possibilities, you will have squamous epithelium. Stratification happens. Stratification means formation of cells into different layers. So based on where you are developing the epithelium, different types of cells will come together and they will form different layers. Skin has 2 different components, dermis and epidermis. Epidermis on the outside and dermis. Both are multiple layered structures. There are multiple layers in dermis and epidermis. Totally 10 layers are there. So each layer has a different function and each layer has a specific structure that is suiting the function. Whatever the function is supposed to be, matching that will be the structure. Alright. Connective tissue, loose connective tissue and dense connective tissue. There are two types of connective tissue, loose connective tissue, dense connective tissue. Loose connective tissue consists of cells, fibers and ground substance. Whether it is loose connective tissue or dense connective tissue, any connective tissue should have these three. There should be a ground substance in which there are cells and fibers. Based on what are the cells and what are the types of fibers you have in that connective tissue, you have different types of connective tissue. Like in loose connective tissue, you will have hyaline, that is cartilage. In loose connective tissue, let's say blood. Blood, are there cells in blood? Is there fibers in blood? What fibers do we have in blood? Albumin, globulin, fibrinogen, those are all fibers, proteins. Do we have ground substance? The plasma is the tissue, stores fat found under skin and around organs. Skeletal tissue, bone and cartilage provide support and structure. You can go and expand further different types of bones, different types of cartilage, cartilage based on the ground substance. Alright, go on, you can elaborate further. Muscular tissue, skeletal muscles, smooth muscles and cardiac muscles. Can you tell me the differences or the structural differences between skeletal, smooth and cardiac muscles? Can you tell me the structural difference between skeletal, smooth and cardiac muscles? The arrangement of the filaments in skeletal and cardiac muscles is similar. The arrangement of the filaments, the arrangement of the filaments is similar. They work as a coordinated unit, one unit. Smooth muscles, the arrangement of filaments is different. They are not in continuity with each other. So smooth muscles can bring about more micro regulation of contraction and relaxation. It can bring about more smaller controls. Skeletal muscles and cardiac muscles will not be able to bring about micro regulation. It will bring about complete regulation. Biceps, when contraction of biceps happens, you will pull this joint. It is meant for that. You cannot have partial contraction of biceps. You cannot have a part of biceps being contracted and rest all not being contracted. That's not possible. But it is possible in your intestines. It is possible in your esophagus, in your stomach, in your intestines. Peristalsis. Peristalsis can happen. A segment is contracting, another segment is relaxing. Though you have continuous sheet of muscles, because it is smooth muscles and the smooth muscles can act independent of each other in spite of being a part of the same sheet. They are a part of the sheet, but the smooth muscles can act in compartments. Small part is contracting, the another part is relaxing. So it allows for pushing of the contents of the tube. So smooth muscles are required for regulating smaller functions in the organs and glands. Skeletal muscles for regulating the skeleton, cardiac muscles for the heart. Alright? Cardiac muscles are present only in the heart and not in any other part of the circulatory system. In all the other parts of the circulatory system, you have smooth muscles. So skeletal muscles, voluntary muscles attached to bones, smooth muscles, involuntary muscles found in walls of internal organs. Cardiac muscle found in heart, involuntary, striated. Striations are because of the arrangement of the fibers in the form of, what is the basic unit of a cardiac muscle and a smooth muscle? Contraction unit. Sarcomeres. The arrangement of filaments, the formation of sarcomeres, the continuity of sarcomeres, thin filaments and thick filaments arranging in the form of bands. Those bands will give you the striations, stripes. Smooth muscles don't have those stripes because the arrangement of fibers is not in the form of sarcomeres. Alright. Nervous tissue, neurons and neuroglia. Nervous tissue has two components. Neurons, which are the functional units of the nervous system. Neuroglia. What are glial cells? Glial cells are the supporting cells in the nervous system. You have different types of glial cells also. Based on the type of glial cells, you have different functions like astrocytes, microglia. All right. Schwann cells are also glial cells. Where do you find Schwann cells? Where do you find Schwann cells? Schwann cells are found in the where? Huh? Axon of a neuron. What do they form? Myelin sheath. All right. Myelin sheath is formed in a concentric layer around round and round. All right. So neurons and neuroglial cells, organs and organ systems. Examples of organ systems: digestive system, respiratory system, circulatory system, nervous system. Different organs. Organs are multiple organs are part of these organ systems. This is very elementary. We will go. This chapter is a summary chapter for what all that you will study subsequently in the rest of the textbook. All right. Morphology and anatomy. Earthworm has a segmented body, segments with setae, clitellum and hermaphrodite. It is hermaphroditic. Hermaphroditism means what? What is hermaphroditism? What is a hermaphrodite? Both sexes or organs, both male and female organs being in the same individual, being in the same organism, that is a hermaphrodite. The same animal can act as a male in one situation and can act as a female in another situation. All right. So that is being hermaphroditic. Digestive system. Complete digestive system starts with mouth and ends with anus. Cockroach. Morphology is segmented body, body divided into head, thorax and abdomen. Nervous system anatomy we will have to study. Simple diagram based study of these systems we will do. Brain, nerve cord and ganglia. See, cockroach, speciality of cockroach nervous system is the ganglia. Ganglia is what makes the distribution of neural regulation decentralized. Alright? Our nervous system is like central nervous system, where all the processing and regulation happens because of the center. The individual organs of the body, the individual parts of the body do not have their own regulation. But in cockroaches, you have ganglia present in the lateral part of the body, throughout the body. Throughout the body there are ganglia. What are ganglia? What are ganglia? I have told this earlier also. Cytons in the? Not in the central nervous system. Cytons outside of the central nervous system are ganglia. A cluster of, you see, clearly identifiable bundles of cytons in the central nervous system is called what? Nucleus. Nuclei. You will find multiple nucleus in the brain. Nucleus with reference to the nervous system is a bundle of clearly identifiable bundle of cell bodies. Alright, you have red nucleus, pyriform nucleus, different types of nuclei you will find in the brain. The same bundles of cell bodies of neurons found outside of the central nervous system is referred to as ganglia. Cell bodies or the cytons are the places of processing in the nervous system. So wherever the cell bodies are present, processing happens. That means what? It can receive information and send messages to action, send messages for action. Because all of that is concentrated in the central nervous system in our body, you chop off the head, the person will die. There is no external regulation, there is no regulation outside of the central nervous system for the body. But in cockroaches, they have a brain, spinal cord, all of that is there. But they have a local regulation of their body functions in the form of multiple ganglia. The cell bodies are present in different segments of the body. So that segment is regulated by this ganglia. You chop off the head of the cockroach, the cockroach will continue to remain alive. It will live. It will die eventually because it will not be able to feed. It will not be able to feed, it will not get food, and therefore it will starve to death. It doesn't die because of the chopping of the head. Alright. See, when you make a glossary, when you note down what is ganglia and one sentence description of what it is, that will give you an understanding of it. It will help you answer multiple questions related to that. Ganglia is a collection of nerve bodies outside of the central nervous system. Ganglia will help in the regulation of functions. That's all. Over, no? Repeated revision of that will give you answers to the questions that are based on ganglia. So if something else comes where ganglia is being referred to, you know that it should be part of the peripheral nervous system. It cannot be part of the central nervous system. Sympathetic ganglia are present in we do have sympathetic ganglia. If the question is sympathetic ganglia are present in option A, midbrain, option B, spinal cord, option C, sympathetic nerves, option D, pons. Which one do you choose? Sympathetic ganglia are present in the four options: midbrain, spinal cord, sympathetic nerves, pons. Sympathetic nerves, because we know nerves are a part of the peripheral nervous system. Nerves are the distribution network. Ganglia, we know by definition, if we know what ganglia is, we know it cannot be a part of the central nervous system. It has to be a part of the peripheral nervous system. And we also know midbrain and pons are a part of the brain, and spinal cord is also part of the central nervous system. So by elimination, just by understanding what ganglia is, you can answer this. Alright? So getting an understanding of different things is very important. Once you have understanding, you should be able to answer any question. Frog, morphology, amphibious, moist skin, anatomy of the frog, circulatory system you will study. Three-chambered heart, closed circulation. Now, there is a condition in human beings also which will make the human heart functionally three-chambered. Which is that condition? Functionally, it is functioning like a three-chambered heart. What is the characteristic of a three-chambered heart? We talk about four chambers in our heart. Which chamber is absent in a three-chambered heart? So there are two atria and two ventricles in our heart. If it is a three-chambered heart, then something is missing. What is missing? One ventricle is missing. That means the reception is happening from both parts. The double circulation is happening. But for both the circulation, it is going from the common ventricle. It is going from common ventricle. Both for pulmonary circulation and for systemic circulation. That means what? Functionally, what is the significance of that? Why is it important to know the limitations of a three-chambered heart? What limitation will it cause? Have you seen frogs? Okay, you have seen cockroach. Cockroach is an insect, arthropod. How many chambers? Does it have a heart? Is there a heart in cockroach? There is no heart. Huh? Insects don't have heart. Huh? There is no circulatory system in cockroach. Circulatory system is there. But is there a heart? Okay, let us look at it. Let us look at chordates only. Amphibia is a part of chordata, no? Amphibia is a part of chordata. Do reptiles have heart? Do reptiles have heart? What kind of heart do they have? Three-chambered heart. Huh? Are amphibians lesser in evolution than reptiles or better in evolution than reptiles? See, think about it logically. Life started to evolve in water. So first, the earliest in the evolution are fishes. Then if you have to get out of water, if you have to get out of water, you have to start developing your ability to live on land also. So after fishes evolved amphibians. Then you can stay in water, but you cannot live in water completely submerged. They are reptiles. Can reptiles stay submerged inside water? Crocodile is a reptile. Crocodile, is it a reptile? Are snakes reptiles? They all can live in water. But can they live inside water completely? They come out to take breath. They have developed mechanisms of slow respiration. They can breathe very, very slowly, so that once they come oxygenate their blood, they can stay inside water for a period of time and come back again to oxygenate their blood by taking in air, so that they can go and stay a little bit more in the inside, on the inside of water. Crocodiles, you see that they put their snout outside of water. For what? Breathing, respiration. Do amphibians run very fast? You have seen frogs. Can the frogs hop very fast? They are slow. See, simple thing is, if the ventricles are separated, if you have two separate ventricles, the advantage is what goes into the systemic circulation is oxygenated blood. Blood which has good amount of oxygen loaded into it. That means it will be capable of supplying the demand of working muscles. It will be capable of supplying the demand of working organs and parts of the body. If the blood that goes into the systemic circulation is partially oxygenated because of mixing with deoxygenated blood also, this ability to supply the demand of oxygen will become lesser, isn't it? Yes or no? So those organisms, those animals which have a three-chambered heart will not be able to do high intensity work. High intensity work is not possible because they will not be able to supply oxygen to the extent of demand. There is one condition in humans also which will make the blood in both the ventricles mix, which will allow mixing of blood in both the ventricles. Which is that condition? Have you studied something called tetralogy of Fallot? Fallott's tetralogy? There are 4 abnormalities. It is a congenital heart disease. One of the abnormalities is incomplete interventricular septum. The septum or the partition between the two ventricles is incomplete. It is not separating the ventricles completely. There is a gap in the interventricular septum. So when the ventricles squeeze blood, ventricles from the left ventricle can go into the right ventricle. Mixing of blood happens, generated in the right ventricle. So when both of them contract simultaneously, the pressure created by left ventricle is higher, so blood leaks into the right ventricle also, making the right ventricle stretch more because it is getting more volume. So the problem here is cardiac output into the systemic circulation goes down. You are not pumping enough blood. It is not blood mixed with deoxygenated blood. It is the flow being reduced. And over a period of time, because the right ventricle is being stretched more than what it is supposed to stretch, and it is pumping more blood than what it is supposed to pump generally, it is undergoing hypertrophy, exercise. It is exercising more. Any muscle which does more work than what it is supposed to do will hypertrophy, meaning it will become bigger in size. Right ventricular hypertrophy is one of the defects there. Alright, so frogs have 3-chambered hearts. Frogs have 3-chambered heart. What is the condition, congenital condition in a human heart which can make human heart functionally 3-chambered? Functionally 3-chambered, not structurally. Can be a question to ask you about tetralogy of Fallot. Tetralogy of Fallot questions can be which of the following are abnormalities found in tetralogy of Fallot. Which of the following is not found in tetralogy of Fallot? It can be reversed also. It can be correlated with this. Or you can correlate that with crocodiles which have incompletely 4-chambered heart. Reptiles have 4-chambered heart, but crocodiles have incompletely 4-chambered heart. That means the interventricular septum is incomplete. So the heart of a child who has tetralogy of Fallot is comparable to the heart of which of the following? Can be a question. The heart of a child with tetralogy of Fallot is comparable to the heart of which of the following? Fishes, amphibians, reptiles, a normal human being. Can be reptiles or crocodile specifically. So question can be anything. Understanding what it is the key. The same thing can be asked in 20 different ways. How many questions do you prepare for? You have to prepare for concepts. So when you are going through all this, lot of cross connection should happen. The entire syllabus map of 11th standard is a little less than 50 pages. But those cross connections should get activated in your brain. You should start thinking, stop doing things mechanically. Don't do things mechanically. There is not enough time. Buck up. Alright, we don't have enough time. Don't be relaxed. Don't get relaxed. Okay? Cell, the unit of life. Cell theory. Historical background is, we have to know contributions by Schleiden, Schwann and Virchow. Schleiden studied the plant cells, Schwann studied the animal cells, Virchow studied the cell division. Questions, if they come on these people, it will be about this. Nothing more. We don't need to know anything more about them. Modern understanding is, all living organisms are composed of cells. All cells arise from pre-existing cells. All cells arise from pre-existing cells. This is the key principles of cell theory. There is a theory of evolution which talks about life originating from non-life. What is that theory? Which contradicts the cell theory. Question can be, which of the following theories of evolution are in contradiction to the cell theory? Darwin's theory, theory of spontaneous generation, exogenesis theory, Lamarck's theory. Which one contradicts cell theory? Spontaneous generation contradicts cell theory, where they say you keep a soiled cloth, you will find some insects coming out of that. You will find maggots coming out of broth, meat broth. So spontaneous generation theory is in contradiction to cell theory. Overview of the cell. Definition: cell is the fundamental structural and functional unit of the cell. Basic units of the cells in all living organisms. Types of cells: prokaryotic and eukaryotic cells. Prokaryotic cells, no nucleus. Eukaryotic cells, nucleus and organelles. I have already told you, functionally prokaryotic cells and eukaryotic cells are similar. There is not much difference between the function of prokaryotic cell and eukaryotic cells. All the functions that a eukaryotic cell does, prokaryotic cells also do. Yes or no? What functions do eukaryotic cells do? There is respiration, there is food breakdown, there is breakdown of chemicals, there is metabolic reactions happening, reproduction happens. All of that happens in prokaryotic cells also. The degree of complexity and the efficiency vary. In eukaryotic cells, the cellular functions happen at a higher level of complexity and more efficiency. Why? Why? Why is there higher level of efficiency and complexity in eukaryotic cells? Or we will talk about efficiency. Why are eukaryotic cells more efficient than prokaryotic cells? Why? We have discussed this. What makes eukaryotic cells more efficient than prokaryotic cells? Okay, formation of cell organelles. Functionally related Functionally related enzymes are all clustered into different organelles. So specific functions happen in organelles which are more efficient than generally happening in the cytoplasm. So eukaryotic cells are more efficient than prokaryotic cells. Alright. Rest of the differences we will see. Structure, basic components of a cell: plasma membrane, cytoplasm and nucleus. Plasma membrane, cytoplasm, nucleus, and in the cytoplasm we have different organelles. Characteristics: prokaryotic cells, small, lack membrane bound organelles, DNA is not enclosed within a nucleus. Cell envelope of a prokaryotic cell. Prokaryotic cell, eukaryotic cell differences with clearly defined parameters and characteristics of both is a table. You should not just copy paste a table that is given in your book. You have to simplify it, make it more easy to revise. Alright? What do you find in the cell envelope of a prokaryotic cell? Glycocalyx, cell wall and plasma membrane, triple layer. Glycocalyx is referred to by two names. If it is thin, it is called slime layer. Slime layer. If it is thicker and harder, it is called capsule. Bacteria with a thick capsule around it are referred to as what bacteria on the basis of staining techniques developed by Gram? Gram? Those with capsule are Gram what? Gram negative. Gram negative. Remember, Gram stain based questions are very, very frequent. Gram staining based questions are common. Biggest confusion is what is Gram negative, what is Gram positive. Gram staining is for cell wall. Gram staining is for cell wall, and bacteria with the capsule do not take up the stain. So you add another secondary stain to stain the capsule. Gram positive are those which will take up the primary stain. If they have to take up the primary stain, they should not have capsule. Bacteria without capsule are Gram positive, bacteria with capsule are Gram negative. Alright? Okay. Ribosomes, 70S ribosomes. Ribosomes are the sites of protein synthesis. Protein synthesis in prokaryotic cells happen with ribosomes as the centers. Even in eukaryotic cells it happens with ribosome only. But ribosomes are organized well on the endoplasmic reticulum to make protein synthesis more efficient in eukaryotic cells. There are inclusion bodies. Inclusions are reserve materials like phosphate granules, glycogen granules. The inclusions are stored in the cytoplasm. Eukaryotic cells, characteristics: membrane bound organelles, well defined nucleus, found in Protista, Plantae, Animalia, fungi, everybody. All other kingdoms except the prokaryotes, except the monerans. Monera is largely represented by bacteria. Cell membrane, phospholipid bilayer. The difference between prokaryotic cell membrane and eukaryotic cell membrane. Cell membrane here is phospholipid bilayer. Even in the prokaryotes it is phospholipid bilayer, explained by the fluid mosaic model. Who gave the fluid mosaic model? Sladen and Schwann. Sladen and Schwann, huh? No? Singer and Nicholson. Okay, Singer and Nicholson gave the fluid mosaic model. Okay. Semi-permeable controls the substance, controls the movement of substances. See, in the semi-permeable membrane, in the fluid mosaic model, you have a phospholipid bilayer and you have proteins embedded in the cell membrane. If you label the proteins with numbers, if you look at a cell and if you look at all the proteins which are present in the cell membrane and label them with numbers, do the proteins remain in their positions throughout the life of a cell? Let's say this is the cell and this, the wall and the roof and the floor is the cell membrane. I have marked the proteins by numbers. This fan is one protein. Let's say it's embedded in the wall. This is protein number 1. Till the life of the cell expires, till the cell is alive, will this fan remain in this position only? Yes or no? Similarly, this is one protein, that is one. Let us say there are 4 proteins on that side, 4 proteins on this side. This is one protein, this is one protein. The lights are all proteins. Everything attached to the wall are all proteins. They all are fluidic in their position. It need not be present at the same place. They can move. They can move in the cell membrane. Their positions are not fixed. Their positions can keep altering. Alright? This part of the membrane could have this part of the membrane can be used up for packaging something to be thrown out of the cell. Then this is no longer available. New membrane would have been added there. The same protein might not have been added. So, fluid mosaic means, it's a mosaic. Mosaic means distribution of so many things, structures. Mosaic. Fluid mosaic means what? It is fluidic. It is not fixed in position. It can keep changing its position. So that is the basic understanding of the fluid mosaic model that you need to have. What is fluid mosaic model? Why is it called so? There are 2 things. What is fluid? Fluid is something that is not stable, that is not fixated. Mosaic means what? Distributed. What is distributed? The proteins in the phospholipid bilayer. So if the distribution, if the mosaic is of the proteins in the phospholipid bilayer, what is fluidic about it? The fluidity is about the position of the proteins. They need not be present in the same place, they can keep moving around. Alright? Cell wall found in plants, algae, fungi, different organisms, and when you are making perspective based notes, cell wall, which organism has what kind of cell wall? Cell wall specific notes you have to make. Composition, what organisms have cell wall? What is the main component? What is the additional components that you find in a cell wall? One small table which lists all the important organisms with cell wall and what the cell wall is made up of. One table will give you enough material to answer all questions related to cell wall. Because you see here, plants have cell wall, algae have cell wall, fungus has cell wall. If we look at prokaryotes, bacteria also has cell wall. So there are 4 different types of organisms having cell wall. Natural question that should come to your mind is, do all of them have the same kind of cell wall? Do all of them have the same structure of their cell wall? Is the composition of all of these the same in terms of the composition? Those questions will give you the table. So the parameters can be structure, composition, uniqueness, whatever, some 3-4 parameters you can have, and all the organisms with different cell walls. So you get a comparison of cell wall between different organisms. The more and more you compare, something is present in many things. If something is present in many things, comparison of all of those things should be in one place. That is a technique. Alright? Only then you will be able to develop a perspective. Endomembrane system includes endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. What is not a part of the endomembrane system among the organelles present in the cell? Mitochondria. Mitochondria is not a part of the endomembrane system. What is the common function that all of these organelles of the endomembrane system are associated with? All the organelles of the endomembrane system are associated with protein synthesis. Yes or no? They are all associated with protein synthesis, related to protein synthesis process. By the end, See, not necessary that all the vacuoles will have proteins. It is not necessary that all the vacuoles will have proteins. But they are associated with Golgi complex in continuity. Golgi complex is getting some instruction and some materials produced in the endoplasmic reticulum. And ribosomes are also part of that. Ribosomes, why should not we consider ribosomes as a part of the endomembrane system? If all of these are associated with protein synthesis, directly or indirectly, why should not we consider ribosomes as a part of the endomembrane system? It is not membrane bound. So endomembrane system is a system of membrane bound organelles which are in continuity with each other. It is a system of membrane bound organelles that are in continuity. Alright? From endoplasmic reticulum to Golgi complex to vacuoles and lysosomes. Lysosomes are also vacuoles, one type of vacuoles. In mitochondria, powerhouse of the cell, site of ATP production, it has a double membrane structure. Why do we need double membrane for mitochondria? There are two organelles in eukaryotic cells which are double membrane organelles. Which are they? As not considering nucleus as organelle. What are the two double membrane organelles? Mitochondria and plastids. What is common to both of these? By which you can correlate their function with structure. ATP synthesis, both of them are associated with electron transport chain. Both of them are associated with electron transport chain. Electron transport chain is dependent on gradient, creating a proton gradient. Electron transport chain is dependent on creating a proton gradient. So double membrane allows flexibility for these organelles to have the space between the two membranes as the concentrating space, where they can concentrate, where they can use to regulate the proton gradient. Otherwise, it is cytoplasm on the outside. What you push outside of the organelle into the cytoplasm can just get distributed, and you will not be able to create a gradient specifically around that organelle. So both of them associated with electron transport chain, both of them need to create a gradient, and both of them have a double membrane structure. Mitochondria has a folded membrane. Plastids don't have folded membrane, inner membrane. Why? Why was there a need for mitochondria to fold? Create folding on the inner membrane. Plastids are associated with transformation of energy from the inorganic world to the organic world. Transformation of energy from the inorganic to the organic world. Correct? On the production of ATP by the plastids, the life of the plant is not dependent. Meaning, the metabolic processes of the plant is not dependent on plastid. It is dependent on ATP production by mitochondria. So mitochondria should be able to produce more ATP to cater to all the demands of all the cells of the body, plant body. Plastids are functional intermittently when there is sunlight available. All the functioning of the plant body is dependent on metabolic reactions leading up to the production of high energy compounds, which needs to be transformed in the mitochondria. Correct? So mitochondria has a greater demand for ATP production, making the requirement for pushing in more and more complexes into the inner mitochondrial membrane. In the same space to push more complexes, you fold. When you fold, you increase the surface area, and increased surface area allows for the mitochondria to embed more and more complexes, electron transport complexes into the system. Okay. All of that has evolved as a response to the challenges to survival. It is all evolutionary adaptations that we see in various of these organelles also. When we look back and try to make sense out of it, we are trying to understand the evolutionary forces that drove it towards the development of those processes. Alright? Understanding? Ribosomes, 80S ribosomes, site of protein synthesis found in cytoplasm and on the rough endoplasmic reticulum. Cytoskeleton is important. They are network of protein fibers including microtubules, microfilaments and intermediate filaments. Microtubules, microfilaments and intermediate filaments. This network of cytoskeletal proteins is what will bring about movement within the cell. If some material has to be transported from one part of the cytoplasm to another part of the cytoplasm, it is not happening randomly. It is happening by the production of these small cytoskeletal elements which will push and pull these components. Whatever needs to be moved within the cell is moved by cytoskeletal elements, microtubules, microfilaments and intermediate filaments. These three you have to remember. When you make perspective based notes, you can make diagrams of this. When you look at the syllabus map, you will understand what can be better explained with the diagram. And you will have all those diagrams in one place. Alright? Cilia and flagella. Cilia and flagella are hair-like structures involved in the cell movement. What is the primary difference between cilia and flagella? Both are hair-like processes. Flagella is for movement of the cell. Cilia is for movement on the surface of the cell. Flagella helps in movement of the cell. Cilia helps in movement on the surface of the cell. So movement of ovum in the fallopian tube is facilitated by cilia and not the flagella. Alright? Okay. Nucleus contains the genetic materials. Genetic material controls cell activities, site of RNA synthesis. Chromosomes, structures within the nucleus containing DNA involved in heredity and cell division. And whenever we talk about this, I want you to recall the sequence of development of chromosomes. First is DNA. DNA coils around what? Histone proteins. So when DNA coiled around histone is referred to as, what do you refer to it as? Louder. Chromatin. Chromatin fiber. Chromatin. You condense this chromatin, it becomes a chromosome. Chromosome. So one unit of condensed chromatin is chromosome. If there are two chromatin threads condensed to form chromosomes, how many chromosomes do we have? Two chromatin threads are condensed. How many chromosomes do we have? Two chromosomes. I bring them together. How many units of condensed chromatin do you have? You have 1 unit of condensed chromatin. It might be 2 chromatin threads coming together, 3 coming together, 4 coming together, doesn't matter. How many unit is there? How many unit is? How many independent masses do you find? 1. You have seen polytene chromosome, giant chromosomes. Giant chromosomes you have seen, polytene chromosomes, they have multiple chromatin threads coming together and forming one giant chromosome. It is still one chromosome. Remember this, this is the basis for answering questions related to cell division. You separate that into 2 units, then you have 2 chromosomes. You bring the 2 units of condensed chromatin and make it into a single unit, you have 1 chromosome. You have 1 chromosome. When you have chromosomes having multiple condensed chromatin threads, how many chromatin threads are condensed to form 1 chromosome here? Two chromatin threads have condensed to form one chromosome. So each one is chromatid. Chromatid. How many chromatids are there in this chromosome? Two chromatids. How many chromosomes do we have? One chromosome. Each chromatid is a representation of one condensed chromatin. So both of them coming together forms one chromosome, and we have two chromatids. You separate the two chromatids. What do we have? What do we have? Two chromosomes. How many chromatids do we have now? We don't have any chromatid. You refer to it as chromatid only when two condensed chromatin threads are together. You understand? Only when more than one condensed chromatin is together, the individual ones are called chromatids and the unit is called chromosome. But when you separate, how many chromosomes do you have? Two chromosomes. How many chromatids you have? Zero chromatids. If the chromatids are of the same chromosome, you call them sister chromatids. If the chromatids are of the same chromosome, you call them sister chromatids. If the chromatids are of different chromosomes, non-sister chromatids. How many chromosomes do we have here? Two chromosomes. How many chromatids? Four chromatids. How many sister chromatids? Two pairs of sister chromatids. Are you understanding? Now I break this. How many chromosomes do we have? Three chromosomes. Three chromosomes. How many chromatids? Two chromatids. How many sister chromatids? One pair of sister chromatids. We don't call these chromatids anymore. Now what do we have? Four chromosomes. How many chromatids? Zero chromatids. Understand? So it's very, very important. This is the basis. You get this, you understand mitosis, meiosis completely. You don't get this, you don't get the difference between mitosis, meiosis. Questions based on mitosis and meiosis are simple, straightforward, but not having clarity on the different stages of appearance of genetic material will lead you to make mistakes. All right. And you have to relate everything logically. There has to be a logical sequence for understanding things. Why it should happen the way it is happening. Then you will know what is not possible. All right. Is separation of genetic material, duplicated DNA, possible before condensation of DNA? Is separation possible before condensation? DNA duplication happens. When DNA is duplicating, is it in the form of chromosome? Does duplication of DNA happen when the DNA is in the form of a chromosome? DNA should be loose. You should copy it, no? Is it easy to make a photocopy of what you have written when the paper is flat and spread out, or when it is folded? When is it easy for you to make a photocopy? When it is neat and flat. That is DNA. You fold the paper nicely into multiple folds, like you make that. Give me one waste sheet, rough sheet. You have not written anything in this. Okay. We will do an activity to understand DNA. Two chromatin threads. All right, two chromatin threads, meaning what? This is DNA plus histone proteins. DNA plus histone proteins. Now, I am duplicating this. I am cutting it into half, but assume that duplication happened. Now I have to replicate this DNA. I have to replicate this DNA. I do DNA replication. I get 2 DNAs. They are copies of each other, exact copies of each other. Now I have to make it into, do it as a chromosome. Hold it like this, back and forth, back and forth. DNA is becoming, what is it becoming now? It is becoming chromosome. What do we have now? Chromosome. What is this? Exact copies. They are exactly similar copies. What do we have? How many chromosomes do we have? 1 chromosome. Now during this is happening when the duplication happened in the interphase. Condensation happened in prophase. Condensation and alignment. Now in metaphase what happens? These fellows will be separated. What do we have here? What is this? Chromosome, chromosome. Then over, that's all. Now when they are together, what do we have? 1 chromosome. How many chromatids? 2 chromatids. What kind of chromatids are these? They are sister chromatids. They are sister chromatids. So during metaphase, metaphase you align. During anaphase, what do you separate? You separate sister chromatids. This is mitosis. Alright. So let us say this is chromosome number 1. Chromosome number 1. We have 2 copies of it. We have 2 chromosome number 1. This is also chromosome number 1. This is also chromosome number 1. Alright. Condense this also. So when 2 people are doing the same task, naturally there is a competitive spirit that comes in, no? Who does it first? Huh? Now he is speeding up. Okay, my chromosome is ready. Chromosome number 1, chromosome number 1. Both of them are aligning in the equator during mitosis. Now in each of them, one goes to the top, both the sister chromatids go to either poles. But in meiosis what happens? This is chromosome 1, this is chromosome 1. Each of them have a pair of sister chromatids. They will not line up next to each other. They will line up like this. So when meiosis 1, anaphase happens, this goes to one side, this goes to one side. So what do we have at the end of anaphase, in telophase? We have a chromosome with a pair of sister chromatids on one side, a chromosome with a pair of sister chromatids on the other side. So we divided the number of chromosomes. We separated the chromosomes, not the sister chromatids in meiosis. In meiosis 2, what happens? this fellow will come and line up in the center, and the sister chromatids are separated. That's all. The difference is who lines up in metaphase and who gets separated in anaphase. That's all. Everything else happens similar. Who lines up in metaphase during mitosis? All the chromosomes line up, one next to the other. That is in mitosis. In meiosis 1, homologous pairs line up. In anaphase of mitosis, sister chromatids are separated. In anaphase of meiosis 1, homologous chromosomes are separated. You get it? That's all. That's all is the difference there. So, when we study chromosomes, structurally we need to study the different stages of development of these chromosomes. Then in the cell we have microbodies, vesicles containing enzymes. Microbodies are vesicles. Vesicles means pouches. Membrane bound pouches containing enzymes involved in various metabolic activities. Are microbodies permanent structures, or can there be temporary microbodies also? Microbodies can be produced from time to time to suit the needs of the cell. Based on the requirement of the cell, microbodies can be produced from time to time. If you want to package a set of enzymes for a particular purpose, you can package it and you can deliver it. That is a microbody. Alright. So it is not necessary that all of them have to be pre-prepared organelles. Organelles are predetermined, pre-prepared organelles. Whatever organelles we study, they are all associated with repetitive and continuous metabolic requirements of the cell. Whatever needs to happen repetitively, whatever needs to happen continuously within the cell, they are organized as organelles in the cell. It is like the parliament. There are committees in the parliament. Have you studied? There are standing committees and there are ad hoc committees. Standing committees are permanent committees. They are associated with the day-to-day functioning of the parliament. There is standing committee on defense, there is standing committee on security, internal security, standing committee on finance. So all of those are standing committees. Suppose a particular bill needs to be discussed. You will make a committee for that particular purpose. That is an ad hoc committee. Ad hoc means temporary, for a particular purpose. Microbodies are like ad hoc committees of parliament, made as per need. Wherever there is a demand, they will be produced. Biomolecules. Analysis of chemical composition. You need to know what is wet weight, what is dry weight, and what is ash. What elements do we find? Chemical composition of biomolecules, inorganic elements, organic compounds. Inorganic elements, what we find in biomolecules predominantly are 4. Carbon, hydrogen, oxygen, nitrogen. You will find these 4 elements in all the living organisms, all the organic matter. Alright. You call that elemental analysis. Whenever you get some fossil or something to study, you will make an elemental analysis of what elements are found. If it is a living organism that is found, whatever remain that you find, it has to have all the 4 elements: carbon, hydrogen, oxygen, nitrogen, which is the fundamental requirement for something to be living on this planet. Carbon, all of them are found in both living and non-living matter. Organic compounds, amino acids, sugars, fatty acids, and nucleotides. These are found in living organisms. Organic and inorganic, the difference is that organic is that which is found in organisms. Inorganic is that which is found in non-living also. Alright? Exclusively found in living organisms is organic, or produced and stored in living organisms. Metabolites you have to study in this chapter: primary metabolites and secondary metabolites. Primary metabolites are those which are produced as a process of metabolism. Secondary metabolites are produced as byproducts of metabolism of the primary metabolites. All right. Amino acids, sugars, fatty acids and nucleotides. Secondary metabolites that you will find is alkaloids. Okay, we'll just look at what they are used for. Amino acids directly involved in growth, development, reproduction. Sugars directly involved in growth, reproduction and development. All of them are associated with all of these functions. Alkaloids, flavonoids, rubber, essential oils. These are all found in plants. Plants produce a lot of alkaloids, flavonoids, rubber and essential oils. Biomolecules are divided into macromolecules and micromolecules. Polysaccharides, nucleic acids and lipids. Structure and function, heteropolymers, structural roles, what catalytic functions they have, structural roles, collagen in connective tissue, catalytic function, enzyme like trypsin. Proteins, types of proteins that we will study, collagen, insulin, trypsin, antibodies, receptors. Structure of proteins, primary, secondary, tertiary structures, quaternary structure. See, each one of these needs to be elaborated. Have you made note of all of these in your syllabus map? Whatever syllabus map you have made? Some of you have done it, some of you have not done it. It has to go to that detailing. Syllabus map is the blueprint. Everything that you expand from your syllabus map will become your level 1 notes. Alright. Structure of protein is complex. If you see the digestion of food in human beings, which nutrient has maximum number of enzymes for digestion? Proteins have maximum number of enzymes for digestion. Proteins are complex structures. The food that we eat will have complex structure proteins. Primary, secondary, tertiary and quaternary structures. You have a simple amino acid chain. Proteins are made from amino acids, no? You have a chain of amino acids, that is the primary structure. You fold it. You fold these amino acids and make it compact, that is secondary structure. That is secondary structure. You make coils of these proteins and make the proteins into balls, where more complex folding and coiling is happening. That is the tertiary structure. You take multiple amino acid based tertiary structures and bring them all together to form one complex structure, that is the quaternary structure. Till tertiary structure, it is all based on single amino acid chain, not single amino acid, one chain of amino acids. One chain of amino acids, plain and simple, is chain structure is primary structure, folded structure is secondary structure, more compacted folding and coiling, coiled structure is tertiary structure. So far the basis of this structure is one chain of amino acids. Now you have another chain of amino acid, bring it to tertiary structure, bring another chain of amino acids into tertiary structure, bring all of them together and make a complex structure. That is the quaternary structure. Quaternary structure. So the proteins that we consume in our diet are quaternary structure proteins. What does our body need? What do we have to convert that into? For our body to utilize it, we have to convert that into amino acids. So you have to break the quaternary structure, make it simple. You have to uncoil it, unfold it, then start chopping off amino acids. There are two types of peptidases, endopeptidases and exopeptidases. If you remember, what are the exopeptidases that you find in your digestive system? Exopeptidases found in human digestive system. What are they? There are two exopeptidases. No? Aminopeptidase and carboxypeptidase. Why aminopeptidase and carboxypeptidase? How is the protein structure? There is an amino group and a carboxy group for every amino acid. Correct? Every amino acid has an amino group and a carboxy group. That is what makes it an amino acid, no? So if you join these amino acids to form a chain, one end of the chain will have a free amino group and one end of the chain will have a free carboxy group. You call that amino terminal and carboxy terminal of an amino acid chain. Aminopeptidase is an enzyme which will break the carb-amino bond, peptide bond. Peptide bond is between carboxy group and amino group of amino acids. Aminopeptidase can break the peptide bond, the first peptide bond, from the amino terminal. So the aminopeptidase enzyme requires a free amino group to attach to. It will attach to the free amino group and then break the first available peptide bond. So what does it do? It will break one amino acid from the chain. From which terminal? From amino terminal. Carboxy peptidase will do the same from carboxy terminal. On the other terminal, it will attach to the carboxy group, break the first peptide bond. So these enzymes are exopeptidases. Means what? Peptidase is what? That which can break the peptide bond. An enzyme which can break the peptide bond is a peptidase. Exopeptidase means what? Exo means outside. That which can break the peptide bond from the outside. Wake her up. Go wash your face and come back. Okay. Exopeptidases can break the peptide bond from external, outside amino acids. Endopeptidases can break the peptide bond anywhere. Any peptide bond can be broken down by endopeptidases. Examples of endopeptidases? What peptidases do you have in the stomach? Pepsin and renin. Both of them are endopeptidase. They can break any peptide bond. In the small intestine we have trypsin, chymotrypsin. They are endopeptidases. They can break any peptide bond. Aminopeptidase, carboxypeptidase are exopeptidases. They can break one peptide bond from the terminal. The last peptide bond is what they can break. The last peptide bond. Aminopeptidase does it from the amino end, carboxypeptidase does it from the carboxy end. So when you are studying biomolecules, you have to correlate that also, because understanding biomolecules will help you understand digestion process. You have to bring the biomolecules from the complex structure to the simplest structure. Complex structure in food is polymeric form. The simplest structure that we need to assimilate is monomeric form. How do we break the polymer into monomer? So how is the polymer formed from the monomers? Reverse of that is digestion, no? So when you study biomolecules, how these biomolecules are produced will give you an understanding to explain how the biomolecule should be broken down to make monomers, which can be absorbed by the body and used. Okay? Understand all that? Okay. Because we are going chapter by chapter, a lot of things are being discussed randomly, in continuity also. This can be a bit overwhelming. I'll give it is 11:12 now. I'll give 7 minutes break. If anybody needs to just take a walk, have some water, if you need to go to the washroom, or you just have to just lie down. Oh, too much now. I need a break. 5-7 minutes break. Water, washroom, sleep. Cool down your brain. He is already jumping with heat in his brain. Seven minutes. 11:20 we start again. We should finish the discussion of the entire syllabus map this week. You have to transfer it. Today after your test, I want all of you to transfer scores of all your previous tests into the booklet that will be given today, the performance analysis booklet, and I want to see the booklet of each one of you tomorrow. All right? You make entries of your performance individually in your booklet. You will know. You will just have to see what is given and you will know what to enter. That is easy. A tabulation of performance. Test by test detailed analysis is there, and in the end, last page, last 2 pages you see in that, you will find analysis of something else. I'll show you. Open this document, come on. Yeah, Gmail. Don't close this, let that be open. Inbox. Refresh. Not connected. No. It's on. You should select connect automatically. So every time it will show. It will connect whenever it's available. See, there is the internet working. No internet access. Wait. Take it from me. You wait, let that be. Browse. Connected late. Yeah. Open that. Open it here only, no problem. No. Yeah, click it. Close that. See, this is the booklet you will be getting. You have to enter your details, you have to write your details. Your name, all your name and relevant personal details you write. For every test, this is what you have to document. All right. How many questions attempted and how many questions are wrong. Subject wise, physics, chemistry, biology. Total questions attempted, total questions wrong. For every subject, what is the negative score, what is the positive score, what is the net score. Same for total you will mention. You understand? That's all. This booklet will have this analysis for every test. For all the 62 tests. Serial number and date is pre-entered. So the same thing for every test. So far whatever you have completed, you will document. This is consolidated progress report. For every test, This is for the overall test, not subject wise. How many questions did you attempt? How many were correct? How many were wrong? Negative score, positive score, total. That's all. This will tell you how you are moving. Alright? We can use this, once you are done, you can use this to graphically analyze also. This is also there for all the 62 tests. So overall you will know, from the first test to the last test, you will know what is the total number of questions you attempted in the entire program. How many did you get it right? How many were wrong? Total negative score that you got, total positive score you got, and what is the total score you have got. So compare it with the total number of questions that you attempted. The overall will tell you what you are prepared for. Alright. So what you get, is it in alignment with your preparation or not, is important. This is the total complete of all the 62 tests. Total questions faced, questions attempted, correct answers, wrong answers, positive score, negative score, total score. You can bring it down to 720 also. I mean, bring it down to 180 for physics. That would be total questions faced. If your score is so much for so many questions faced, what would your score be for 180 questions faced? Not 180 questions, 45 questions, 180 marks. Same for chemistry, same for biology. Total questions faced. That is, what is the total number of questions that you got in the form of question papers? Total questions attempted, total questions correct, total wrong, positive score, negative score, total. This document will be your lifetime record. Alright. This is what I wanted you all to do, but you are not doing it, so I am giving it. Alright. And as usual, I will give some quotes of mine, which I have given here also. The right process leads to the right results. Trust in your abilities to take you through all challenges. You are capable. Alright, that's all. So, this you will get, because I want you to complete this today. Only of those tests that have been completed. It is all self-explanatory. You just have to look at it and you have to enter it. I hope that all of you have written down your test performance details in one place. Have you or have you not? I told you what to document before the first test only. So all of that should be documented. All right. So we'll go ahead with this now. See, here quaternary structures. Primary structure, sequence of amino acids. Secondary structure, alpha helix or beta sheet, just single folded structure. Tertiary structure, three-dimensional structure like a ball. Quaternary structure is multiple balls put together. That's all. Polysaccharides, cellulose and starch. Cellulose, starch and glycogen are the three types of polysaccharides. Cellulose is structural component of plant cell walls. Starch is energy storage in plants. Glycogen is energy storage in animals. Function of polysaccharides, energy storage. Structural components forms the structural components. Cellulose in plant cell walls. Nucleic acid, types of nucleic acid, DNA, RNA. DNA is double-stranded, carries genetic information. RNA is single-stranded, involved in protein synthesis. RNA also functions like enzyme in certain situations. Ribozyme is an enzyme. Ribozyme is an enzyme involved in protein synthesis. Ribozymal enzymatic action. You will find it in protein synthesis. Otherwise, enzymes, ribosomes are all for translation process. Structure of nucleic acids, nucleotide structure composed of sugar, phosphate and nitrogenous base. All of this is very simple. Sugar, phosphate, base. That's all. Once you know it, it is simple. These are the small things that will take you wrong, that will make you go wrong. So basics, fundamental things, foundational things should not be ignored, should not be overlooked. Oversight, errors are blunders. Oversight errors are what will pull you down. You don't get some complex question and you try it and you get it wrong, those are fine, they will not pull you down. Oversight errors will pull you down, and oversight errors happen with easy questions. Easy questions are the ones which will bring down your performance, because you are more likely to commit oversight errors with easy questions. You become overconfident. When the paper starts appearing easy, you become overconfident. In overconfidence, your mind will tell you to do all the easy things very fast, so that you will get more time to solve the difficult things, difficult questions. So in the drive to get the kick of solving complex questions, you will rush with easy questions, putting you at risk of getting simple questions wrong. Oversight errors are blunders, remember. And oversight errors will not happen if you are respectful of your syllabus map, your level 1 notes, and your perspective based notes. These three will help you not commit any oversight errors. All right. DNA, RNA structures we saw. Nucleotide structure, sugar phosphate backbone provides structural framework for nucleic acids. DNA structure contains deoxyribose sugar and a double helix, and its double helix. RNA structure contains ribose sugar and single stranded. Here the base structures you should know. Why adenine will pair with thymine only, and CG, cytosine and guanine. Why that pairing? So structure, single ring structure, double ring structure, those structures also you need to be aware of. Do you still have biomolecules in chemistry also? You have biomolecules in chemistry. Study biomolecules from biology along with biomolecules from chemistry. Study that together. Enzymes, nature and functions. Most of the enzymes are proteinaceous, made of proteins, highly specific. Catalytic power increases reaction rate, lower activation energy. Activation energy is a concept that you need to study well. This activation energy is useful in understanding what process this? Biotechnological processes, where we fermentation process and all that, where we produce, what do we call that? Anyways, leave. Activation energy concept, get strong with it. Factors affecting enzyme activity. Temperature. Enzymes have optimal temperature. See, higher temperature and lower temperature beyond the range of the enzymatic optimum. Enzyme has an optimum temperature, so lesser than that or more than that, both are not good for the enzyme. Lowering the temperature will make the enzyme inactive. Highering the temperature, increasing the temperature beyond a certain limit will make the enzyme get denatured. Why denaturation? Because enzymes are proteins and heat can denature proteins. Denaturation can happen with high temperature. pH. Enzymes have optimal pH. Substrate concentration affects the rate of enzyme activity. Inhibitors, chemicals that reduce enzyme activity. Substrate and, see, in living systems, almost all enzymes, or we can safely say most enzymes, are bidirectional in catalysis, meaning they can catalyze reactions in both the directions. What determines the direction of enzymatic activity for enzymes? What determines the direction of enzymatic activity? Concentration of the reactants. Concentration of the reactants. For an enzyme, that which is higher in concentration is the substrate, and that which is lower in concentration is the product that it will facilitate in the production. Alright? So there is no substrate and product for an enzyme in biological systems. Whatever is higher in concentration is the substrate, and the transformed product, transformation outcome is the product. Once this becomes more in concentration, enzyme will look at that as the substrate. this will be more. So with enzymatic action, the concentration of substrate is always higher. So, or if we talk about reactants in an enzymatic reaction, that which is in higher concentration is the substrate. Concentration of product can also increase. When that happens, the enzyme will reverse the direction of catalysis. Alright? Classification of enzymes are important. Classification of enzymes is important. Oxidoreductases catalyze oxidation reduction reaction. Can you give me one example of an oxidoreductase from glycolysis Krebs cycle? Oxidoreductase. NADH dehydrogenase is an oxidoreductase. NADH dehydrogenase is removing electrons and pumping electrons into the electron transport chain, complex 1 of electron transport chain. Isn't that an oxidoreductase? Catalyzing oxidation reduction reaction. See, enzymes will be given, different enzymes, not outside of your syllabus. Enzymes that you will encounter in your syllabus will be given, and you will be asked to identify the nature of enzyme. So, making a list of all the enzymes that you have to study in your book. While making perspective based notes, you can make a table of all the enzymes. Table of all the enzymes. It can be the name of the enzyme, in which pathway do you find the enzyme, what type of enzyme it is, what is the substrate, what is the product. That's all. You can even add energy dependency. You can even add energy dependency or energy outcome as one of the columns where you can mention whether energy is being used or energy is being produced. So some 5-6 column, a list of all the enzymes. That will cover all the biochemical reactions that you will have to study. Alright. So you have 10 enzymes in glycolysis, 8 enzymes in Krebs cycle, some enzymes of photosynthesis. Where else do you have enzymes? That's all, no? Enzymes in digestion. In human physiology, you have enzymes in digestion. In biotechnology, in microbes in culture and all those things, all those places in your 12th standard syllabus, some enzymes you will find. So if you put together all the enzymes in one place, you might have to study about 50 to 60 enzymes total. One table? Good, no? So are you understanding all this? Put it all together, perspectives. As you are studying, you can make note of that. You can make note of this in this suggestion that I am giving you now. As I am discussing, you should make note of those things. Make a table of all, comparative table of all the enzymes. So if questions come on the nature of enzymes and the type of enzymes, what type of enzyme it can be, one table can save you. So oxidoreductases, transferases, transfers functional group between molecules. Can you give some examples? Transferring functional group between molecules, which will shift the functional group from one to another. Are kinases not transferases? Kinases, let's say phosphofructokinase, is it not a transferase? It is removing the functional group, that is phosphate group, from ATP and transferring it to fructose 6 phosphate, making it fructose 1,6 bisphosphate. That is also a transferase, no? Then hydrolases, catalyze hydrolysis reaction. Hydrolysis is what? Any reaction where water is removed as a byproduct. Any reaction where water is removed as a byproduct. You will find reactions in Krebs cycle where water is removed as a byproduct. Alright. Then lyases, catalyzes the breaking of bonds by means other than hydrolysis and oxidation. Correct? These spam calls They continuously call. Okay, I'll keep it on. Lyases catalyze the breaking of bonds by means other than hydrolysis and oxidation, like enolase. Phosphoenol pyruvate is broken down to pyruvate by enolase. Enolase, that is kinase, phosphoenol pyruvate kinase. Enolase is 2-phosphoglycerate being converted to phosphoenol pyruvate. Correct? So, no, that is also again kinase there. Pyruvate kinase also functions like a lyase, breaking down phosphate from the phosphoenol pyruvate. So lyases breaks down bonds without oxidation or using water, hydrolysis. Isomerases, catalyze isomerization changes within a single molecule. Ligases join two molecules together. Direct isomerases are there in glycolysis. Which are those? Phosphotriose isomerase, which isomerizes between glyceraldehyde 3-phosphate and 1-glyceraldehyde 3-phosphate and 1,3-bisphosphoglycerate is broken down into two compounds. What are the two compounds that it is broken down into? DHAP and G3P. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. All forgotten. Gone. See? Importance of revision. When I taught you glycolysis, Krebs cycle, electron transport chain, all of you could understand and repeat. You repeated the reactions with enzymes, energy, dynamics, everything. Within 2 weeks, it's all gone. Some of you are asking me, we should go chapter by chapter discussing all the concepts in detail so that we will learn it well. You learnt it well. Why are you not able to remember? Problem of not being able to perform well in exams is not learning it well. The problem is because not registering it and not developing mechanisms of recall. Alright? Understanding happens when a teacher teaches you something. When I teach you something, you will understand. I open up the understanding. You have to seal it with revision material. And the revision material is the notes that you are making, which you have to revise. Alright? Most mediocre people take task-based approach. All excellent people, successful people take purpose-based approach. What is it? Task-based approach versus purpose-driven approach. Task-based approach is you are told to do something, not doing which will lead to some consequences. You are expected to do something, not doing it will lead to consequences. Consequences can be punishment or humiliation or loss of something that you hold, something that you will lose. That is consequence. So you do it in such a way that you don't face the consequence. That is task-based approach. Go back to your entire student life. How much of your studies has been task-based? You do homework given by your teachers in school. Why? Because you love doing it, or not doing it will have consequences the next day. Not doing it will have consequences the next day. You will be punished, you will be beaten, you will be scolded, you will be made to stand outside the class. Correct? No homework is given on a particular day. There is nothing which will have consequence for the next day. How much time do you give for studies? Go back into your own student life. How much time would you give to studies if there are no consequences the next day? There will be nothing that will have any consequence the next day. So, you don't touch your books, right? The biggest challenge for you all here in these few months of training that you are going through is to transition from task-based approach to life to purpose-based approach to life. So if it is a purpose-driven approach, as a student, your purpose is supposed to be knowledge acquisition. You have to gain knowledge. You have to gain understanding. You have to learn the ability, you have to develop the ability to transform data into information, information into knowledge, knowledge into wisdom, and wisdom into solutions for problems in life. That is the transition that needs to happen. That is the ability transition that needs to happen through education. But for the entire duration of schooling, college, graduation, post-graduation, most of the people, when I say most of the people, I will be very kind and generous and say more than 95% of the people. I am being kind and generous. While saying that more than 95% of the people complete their entire so-called education with a task-based approach. Go and observe what people in your known circles who are doing PhDs are doing. They would have had 3 years, 4 years to complete their research and prepare their thesis. Before the final submission of their thesis, last 5 to 6 days will be super aggressive work. They will be just going through, putting together some things in a super pace and somehow trying to submit their thesis and complete it. Over, done. Task is complete and the reward for the task is the certificate. But life rewards you for your abilities, not for your certificates. All right. We have not yet completed 2 months of the program. Transform the way you look at life. It is not task-based. Task-based is when you do things when somebody is watching you. You do things if you will be at a loss if you don't do it. Purpose-based is you do it because you know why you should do it. All right. So that transition, it's not the end of your education. Education continues for a lifetime. So you need to constantly work on making yourself capable of transforming data into information. What is the difference between data and information? I give you some random set of numbers, that is data. I make sense of data, that is information. I tell you what that data is about. I'll tell you this is the score of 10th standard students of a particular region in their final exams. Then the number will start making some sense. Sensible data is information. When that information gives you insight, that is knowledge. Then you understand that so many people have performed it in a particular way, so many people have scored so much. That is knowledge. Wisdom is when you start understanding the cause and effect relationship. You understand what is the cause, what is the reason for that kind of a performance. What has led to this kind of a performance. That is wisdom. You understand it and you figure out the cause and effect relationship of your knowledge. The next stage, which will give you money, reward, recognition, status, everything, is your ability to use that wisdom to solve problems. All right? To solve the problem of low performance. Centers, back-end customer support. Correct, no? MSc and PhD, working in college for 15,000 rupees per month salary. Task-based. All right. So my entire focus is ability driven. You have the potential has to be transformed into abilities. Abilities will help you perform whatever you want to perform. Okay. If you understand, so when you have wisdom, when you are wise and when you know how to understand and solve the problems, most of the people are working in a task-based approach. If you look at how your program is going on, the Star 30 program, the policies, processes and systems of the Star 30 program, the approach of the Star 30 program, and what is being done by most of the institutes which train students for NEET and other entrance exams, you'll clearly see the difference. Task-based to purpose-based. In task-based, success is measured in statistics. In purpose-based, success is measured in outcome. What is the difference between statistics and outcome? 5 lakh students, 5 students in top 10, success. Outcome-based approaches, 5 lakh students, what happened to each of the students because of that particular program. Not all the 5 lakh students will get into a medical college because it's impossible. The number of medical seats are limited. But if 5 lakh students have come to you for a particular period of time to get trained on something, the outcome of that training, if it is only a medical seat, most of the students fail in that because everybody will not get a medical seat. Clear. But what did this entire period of training do to them will determine whether your process has been good or not. Alright, so that is how education systems should be designed. Unfortunately, all of us, everybody have gone through the task-based approach to education, making individuals machines. Task-based approach will make you mechanical. Purpose-based approach will make you intellectual. Alright, think. Don't do anything mechanically. Ask questions. Keep asking questions. Try to find out why something is there the way it is. Alright? Keep working on that. We have time. We have time, but we don't have time. We have enough time for sincere preparation and the right approach-based preparation to give you outcomes. We don't have enough time to be complacent. There is no time for complacency, but there is enough time for sincerity. Alright, so lyases, isomerases, ligases joining together. See, if you look at it, oxidoreductase tells it, it's a self-explanatory term, oxidation reduction. Transferase, transfer something, hydrolase. Hydrolase and lyase are similar enzymes which will break. Hydrolase will break using water, that's water-based catalysis, hydrolysis reaction. Lyases without using water or oxidation. Isomerase shift, move between isomeric forms. Ligases joining. Cofactors, prosthetic groups, coenzymes and metal ions. Prosthetic groups tightly bound to enzymes necessary for enzymatic activity. Coenzymes loosely bound to enzymes, often vitamins. Metal ions act as enzyme activators, example magnesium and zinc. The relevance of vitamins in human body is vitamins are predominantly functioning as coenzymes for enzymatic activity. The enzyme which is necessary for the production of rhodopsin pigment in the eye is dependent on the vitamin A. If vitamin A is not available, the enzyme's efficiency will come down. If the enzyme's efficiency comes down, production of rhodopsin will become reduced. When that decline in the production of the product of the enzyme activity, in this case rhodopsin, is so low that it affects the function, the person will start experiencing difficulty in night vision, eventually leading to night blindness. That is vitamin A. It is not like a switch. There will always be indicators, especially metabolic changes. There will be indicators. If you are not having enough calcium in your diet, you will have indicators of not being able to perform muscle activities properly, running and all those things. You will have cramps. You will have cramps during resting stage. Calf. If your calf muscle is cramping, that is a very clear indication that you are having calcium deficiency. You have to increase calcium intake in your diet. Okay. So metabolic changes will always give you indications. Cell cycle and cell division. In our different discussions, we have discussed a lot about cell division, so we'll just rush through. Cell cycle, phases of cell cycle, interphase, M phase. Interphase and M phase or mitotic phase are a part of a dividing cell. In interphase you have G1, S, G2 phase. In mitotic phase you have prophase, metaphase, anaphase, telophase. Then cytokinesis. Karyokinesis, cytokinesis, two things, and goes on. There is a G0 phase also. What is G0 phase? That is the phase of metabolic activity, where the cell is not going through division. In the interphase, the cell is being initiated into division. In the mitotic phase or in the M phase, the division phase, the cell is undergoing division. G0 phase that is the metabolic phase, where the cell is performing its normal function, not going into division. That is G0 phase. G1 phase, interval between mitosis and initiation of DNA replication. S phase is the synthetic phase, DNA synthesis or replication. G2 phase, proteins are synthesized and cell growth continues. Questions have come on what is replicating in G1, S and G2 phase. Different organelles will be given. What replicates in which phase? Protein synthesis is in G2 phase. Important. Then without protein synthesis, how are the enzymes produced in G1 phase? For DNA replication, you need to produce histone proteins, no? You are replicating new DNA. You are replicating the DNA and producing new DNA. For that new DNA to be converted into chromatin for further processing, you need proteins. Protein synthesis should happen in G1 also. Protein synthesis continues in G2. Okay. So G1 phase, cell is metabolically active and grows continuously. S phase, amount of DNA per cell doubles. G2 phase, preparation for mitosis. This we have already discussed. Prophase, metaphase, anaphase, telophase. Prophase, DNA replication has happened. From there to condensation of DNA completion, till the DNA completely condenses. Chromosomes are formed. When do you start seeing chromosomes? End of prophase, end of metaphase, end of anaphase, end of telophase. When do you start seeing chromosomes? End of prophase. At the end of prophase, you see chromosomes. At the end of anaphase, you don't see chromosomes. End of anaphase is similar to beginning of prophase. End of anaphase is similar to beginning of prophase. Alright. So in telophase, no, anaphase is complete when the nucleus separation is complete. Beginning of telophase you see chromosomes. End of telophase is like beginning of prophase. End of telophase is like beginning of prophase. In the beginning of prophase, you don't see chromosomes. Diffuse chromatin network is seen. Okay, we'll do this. All right. Prophase beginning, prophase end, metaphase beginning, metaphase end. Then we have anaphase beginning, anaphase end, telophase beginning, telophase end. You look at these two together. In the beginning of prophase, DNA condensation begins. In the end of telophase, DNA decondensation, chromosome decondensation is complete. So in both these, you don't see chromosomes. In the end of prophase, you see condensation of chromosome complete. In the beginning of telophase, you see chromosomes still present. All right. In the this is not correlating. It is in continuity. These two you have to look at it. Beginning of prophase and end of telophase is similar, where you don't find chromosomes. Questions can be based on which are the phases where you can see chromosomes. Where can you see chromosomes? In which phases do you see chromosomes clearly? In metaphase and anaphase. In metaphase and anaphase, you see chromosomes clearly. All right. By the end of anaphase, chromosomes are separated to the opposite poles. In telophase, nuclear membrane forms. All right. Here, nuclear membrane starts forming in the beginning of telophase. Nuclear membrane is completed in the end of telophase. Here you will still find nucleus. In the beginning of prophase, nucleus is visible. Nuclear membrane starts disintegrating and becomes completely disintegrated, and chromosomes are formed here. Here chromosomes are visible, nucleus is not formed. So by the end of it, nuclear membrane is formed. What happens from the beginning to end of prophase reverses from the beginning to end of telophase. That's all. You have to go back, no? You have to go back to what it was. So chromosomes are clearly visible when? Chromosomes are clearly visible if phases are mentioned: prophase, metaphase, anaphase, telophase. If all the four phases are mentioned, chromosomes are clearly visible in metaphase and anaphase. If the stages of every phase is mentioned, are chromosomes visible at the beginning of prophase? You just say yes and no. Beginning of prophase, no. End of prophase, chromosome visibility, yes. Beginning of metaphase, yes. End of metaphase, yes. Beginning of anaphase, end of anaphase, yes. Beginning of telophase, yes. End of telophase, no. So in all the phases of karyokinesis stage of mitosis, in the beginning of prophase and the end of telophase, you don't find chromosomes. You are not able to see chromosomes. In rest all stages, you will see chromosomes. Metaphase, anaphase, throughout you will see chromosomes. All right. Chromosomes are clearly defined and arranged in which phase? Metaphase. Cytokinesis, division of cytoplasm. Plant cell and animal cell, there is a difference in cytokinesis. In animal cell, there is furrowing of the cell membrane. In plant cell, there is formation of cell plate formation. That question based on that has reduced. Earlier that used to be a repeating question. Now it is not appearing so much, but that's a very basic difference that we need to know. Significance of mitosis: growth of multicellular organisms, cell repair. Meiosis: reduction, division, 2 sequential cycles of division, formation of 4 haploid cells. So each phase has been given. I have already told you. Where do you find the difference? Tell me the metaphase difference between mitosis and meiosis 1. Metaphase difference. In mitosis, lining up of all chromosomes. In meiosis 1, lining up of pairs of homologous chromosomes. Alright, that's all. In anaphase, difference between mitosis and meiosis. Anaphase difference. Anaphase, separation of sister chromatids of every chromosome in mitosis, separation of homologous chromosomes in meiosis 1. Alright, that's the difference. So I have already told you in cell division, it is not only the number of chromosomes that we have to look at, it is also the content of DNA. How much DNA? Double reduction should happen. One is reduction in meiosis, reduction in the number of chromosomes and reduction in the content of DNA, quantity of DNA. How much DNA is present? Both should be half compared to the parent cell. Whereas in mitosis, both the number of chromosomes as well as the quantity of DNA is exactly same as that of the parent cell. Significance of meiosis, maintenance of chromosome number, genetic variability. Photosynthesis in higher plants. What is definition of photosynthesis? Synthesis of organic compounds using sunlight. That's all. Early experiments. If at all questions come about scientists, it will be about this. Joseph Priestley, he did the experiment where he discovered the role of air. Plants restore oxygen that animals remove from air. Air is essential for plant growth. Closed jar experiment with plants and candles where oxygen is used up, carbon dioxide is increased. And Ingenhouse's experiment, role of sunlight in photosynthesis, oxygen produced only in the presence of sunlight. Aquatic plants experiment, oxygen bubbles observed in sunlight, absent in the dark. So those experiments, remember those experiments. Engelmann's experiment, light dependent reaction discovery. First identification of action spectrum. See, action spectrum is which light in which wavelength is more productive for photosynthesis. Where does photosynthesis occur? Chloroplast. Structure of chloroplast, grana, stroma and membrane. Functions of that also we need to know. Grana is the site of light reaction. Simple straightforward questions. The site of light reaction is grana, stroma or the membrane. Direct, outright. Grana is the site of light reaction. Thylakoid membrane houses the photosystems. What houses the, what is equivalent to the photosystem in mitochondria? What we call as photosystems here is for what? Photosystems are the ones which are responsible for electron transport chain. Photosystems are equivalent to our mitochondrial complexes. The photosystems are the ones which will bring about electron transport. Electron transport is happening in the photosystems. Where do we find the equivalent of photosystems in mitochondria? Where do we find the equivalent of photosystems in mitochondria? On the inner mitochondrial membrane. So the electron transport chain photosystems, or not photosystems, the complexes are embedded in the inner membrane of mitochondria, that is on the membrane of the organelle. Here photosystems are embedded in the thylakoid membranes of grana. Do all of you know what thylakoid is? No? Forgotten? Double membrane, chloroplast. Thylakoids are this is the grana. These stacks of plates. The stacks of plates that you find in the chloroplast is the grana. What you find in between is the stroma. Thylakoids are individual plates. The individual plates in the stacks of plates are the thylakoids. The photosystems are embedded in the thylakoid membrane. Thylakoid membrane. So, if these photosystems are bringing about electron transport chain, what are they supposed to create? What are they supposed to create? For electron transport chain to function, there is a need for what? Proton gradient. Proton gradient. Across what will the proton gradient be produced now? Proton gradient will be produced across the thylakoid membrane. Correct? It has to be produced across the thylakoid membrane. So either you push protons from inside to outside or outside to inside. Which one is more conducive? Outside to inside. Why? Thylakoid is a small space. Bringing in something to a smaller space increases gradient. If you push protons from inside to outside, what will happen? They will get diffused into the entire stroma of the chloroplast or plastids. So when you pull out protons from the stroma and push it into the thylakoids across the thylakoid membrane, relative to that, around that membrane, across that membrane, there is more protons inside and less protons outside, creating a gradient. Yes or no? So here, movement of protons will happen across what for the production of ATP? ATP synthase will have movement of protons from where to where? From inside of the thylakoid to the stroma. From the thylakoid to the stroma. That is the movement. From inside to outside, protons flow, making the ATP synthase to move and produce ATP. In mitochondria, it is from outside to inside that the protons move. See, even in mitochondria you see, for creating gradient, where are we pushing the protons? Where are we pushing the protons in mitochondria? From inside, that is the matrix, to the intermembrane space. Which one is a bigger compartment? Is the intermembrane space bigger compartment or the matrix of mitochondria? This is a bigger space. So if you want a gradient across this membrane, where do you want to increase the proton concentration? Here. If you increase the proton concentration in the intermembrane space, at any point of the membrane, at any place along the membrane, you have more protons here than here. Because whatever protons are present, pushed into this will get diffused in this entire space. Correct? Let's say it's like having people. Along this wall, how many people are there depends where people move. Consider protons. Once people move out, they will balance according to the density. Like there are 48 people here. 48 people here. You are supposed to be present in this room. So you arrange yourself to ensure that each one of you have same space around you. That is diffusion principle. That is the principle of diffusion, no? So that everybody has space equally around yourselves. If you go outside also, the same thing will happen. Even when you go outside, you will distribute yourself according to the space available. Correct? Where is a chance of more people being in a given space? If I say in a 10 square feet area, in a 10 square feet area, how many people are there on the inside and how many people are there on the outside? Where do you find more people? In a 10 square feet area, if I say, where do we find more people? Inside this room or outside this room? Outside, huh? Inside, because this is a small space and the area available is less. So even if you rearrange yourself by diffusion, there are more people in any given area compared to outside, which is very vast. Very vast. So if you want to create a gradient, similarly here, if you have, let's say, some x amount of protons here and x amount of y amount of protons here, all the protons present in the matrix of mitochondria will distribute according to the space available. There is more space available, so more protons will get distributed farther from each other. So if I take any particular space here, the number of protons available will be limited. But you pump in a lot of protons into the intermembrane space. So in any given space, in the intermembrane space, the number of protons available will be more compared to a similar space in the matrix. Are you understanding? If we consider 1 square millimeter area or 1 square micrometer area of the matrix versus 1 square micrometer area of the intermembrane space, there will be more protons in 1 square millimeter area of the intermembrane space, micrometer area of the intermembrane space, compared to 1 square micrometer area of the matrix. Yes or no? Because of the space itself. Gradient creation is easier. You have to create gradient to make the protons flow. That will allow you to produce ATP. But gradient, in which direction will you create? Will you create a gradient from a bigger space to a smaller space, or from a smaller space to a bigger space? Which one is more conducive for creating a gradient? Smaller space to bigger space is easier to create a gradient. So what do? You will start loading up protons into the small space to increase its concentration. When you load protons into the smaller space, concentration becomes bigger. It is like putting 1 spoon of salt in 100 ml of water and 1 spoon of salt in 1 liter of water. Which one will be more concentrated? Understand? 1 spoon of salt in 100 ml of water, this is more concentrated than 1 spoon of salt in 1 liter of water. So much needs to happen, so much has been developed by trial and error through evolution. This too, the mechanism of gradient creation in mitochondria and in chloroplast during photosynthesis, during respiration and photosynthesis respectively, will be explored extensively for questions. You have to give a thought to it. You have to develop in your own thinking, in your own words, explanation for this. When I tell you, you might understand. You might not understand also. But this understanding should come in your own words. Alright? Is it clear? So here gradient is across the membrane of the organelle. Here gradient is across the membrane of the thylakoid. Here it is from outside to inside for ATP production. Here it is from inside to outside for ATP production. First thing, smaller space to bigger space is the movement during gradient dependent flow. Gradient dependent flow. Gradient dependent flow is what will produce ATP. For creating gradient, movement is from bigger space to smaller space. To create a gradient, you will move the substance that you want to concentrate from a bigger space to a smaller space. Alright? So here gradient is across the inner membrane of the mitochondria. So bigger space is matrix, smaller space is intermembrane space. To create gradient, you will move protons from inside to outside of the inner membrane. For ATP to the stroma. Clear? Such small things, inside, outside, outside, inside, gradient creation, gradient dependent movement. Alright. This is all, the mitochondria and plastids, electron transport chain, electron transport chain, and that happening in these two is a good content for a drama. If it is in a 11th standard class, a regular class, time can be given for students to make a drama on this. Alright. So you have some very complex intellectual drama you can create. Audience will get, they'll see moving from here to here, here to here. You will have a color code. Alright. Protons will be in some color t-shirt, basically. Membrane, bigger space, smaller space. All of this can be beautifully explained through a drama. But now we don't have time for drama. We are going with the basic assumption that all of you have learnt all of this, which is a wrong assumption. Okay. Okay. So grana is the site of light reaction, stroma is the site of dark reaction. In stroma you find the enzymes for Calvin cycle. Chloroplasts are found in the mesophyll cells. Chloroplasts are concentrated in the palisade parenchyma of the leaves. Pigments, chlorophyll A, B, xanthophylls, carotenoids. Chlorophyll you have A, B, C also. Chlorophyll A is the primary pigment, absorbs red and blue light most effectively. Chlorophyll B and xanthophylls, accessory pigments, captures light and pass energy to chlorophyll A. Important. This we will understand when we study the photosystems. Alright. You have all those antenna complexes, you have the core. It's a beautiful mechanism of energy transfer. To excite electrons, so much coordinated action happens. Next class, tomorrow we will, no, not tomorrow, we will complete the entire syllabus map and then we will come back. But it's a beautiful game of energy transfer, cooperation and collaboration. What can cooperation, collaboration and coordination do? You can see in the light reaction of photosynthesis. Alright, 3 C's for a successful life. If you want to become successful in business, in your social life, even in family, 3C. Cooperation. Cooperation increases efficiency of performance for everybody. Collaboration increases the productivity for the team. Coordination increases effectiveness and reduces time of performing action. Coordination, cooperation, collaboration, coordination. 3 C's. You build institutions, you build organizations, you build companies, whatever you build, you should work on establishing these 3 C's within your teams. People should cooperate, collaborate and coordinate. There is one more C which will destroy you if it goes into your head. What is that? Which you are feeling about each other. Competition. If you feel competition, you will die. Okay? Die literally also, if it gets too much into your head, and metaphorically also, because competition, a sense of competition is what will make you underperform. Though people tell that competition will get you motivated. Again, task-based approach. If it is purpose-based approach, your continuous effort is to make yourself better. You are the center of focus in a purpose-based approach. You are working on competency enhancement. In a task-based approach, certificate drives you. Alright. So, take care of 3 C's, develop 3 C's, ignore 1 C. Ignore competition. Don't bother. Alright. You can anyways not control it, unless you have some mechanism of eliminating competition. That will come in later. Elimination of competition. Light reaction, you have 2 photosystems, photosystem 1, photosystem 2, and there is photolysis of water happening. Photosystem 1, chlorophyll A absorbs 700 nanometer wavelength. Most important critical components of each of these things are given in this one. Photosystem 1 produces NADPH in non-cyclic photophosphorylation. Photosystem 2, chlorophyll A absorbs 680 nanometer wavelength. These two are repeated questions. What is the wavelength of light associated with each of the photosystems? Photosystem 1, 700 nanometer, photosystem 2, 680 nanometer. Water splitting happens in photosystem 2, releasing oxygen. Photolysis of water, splitting of water into hydrogen, electron and oxygen. Electrons replace those lost in photosystem 2. We'll come there. Somebody is losing electrons, somebody is giving electrons. Electron source should be there, electron donor, electron acceptor. That is the basis of electron transport chain. Alright. Z scheme, electrons passed from photosystem 2 to photosystem 1. Z scheme of electron transport. Energy used to pump protons and generate ATP. Principle is the same. Electron transport chain is the principle on which this will drive. Cyclic photophosphorylation, only photosystem 1 is involved, produces only ATP, no NADPH produced. Non-cyclic photophosphorylation involves both PS1 and PS2, produces both ATP and NADPH. You should know where cyclic photophosphorylation happens and where non-cyclic photophosphorylation happens. What conditions will lead to cyclic and non-cyclic photophosphorylation. Calvin cycle is for carbon fixation. Carbon dioxide is fixed into a 3-carbon compound. Catalyzed by Rubisco. What is Rubisco? Expand ribulose, bisphosphate, carboxylase, oxygenase, not oxidase, oxygenase. Got it? Reduction, carbon fixation, reduction and regeneration of RUBP. In reduction, ATP and NADPH used to convert 3-phosphoglyceric acid into glyceraldehyde 3-phosphate. 6 glyceraldehyde 3-phosphate molecules are produced, 1 is released. Right. Regeneration of RUBP. RUBP is regenerated using ATP. 5 G3P molecules recycled to regenerate RUBP. All of this important, the entire cycles we have to remember. C4 pathway happens in the mesophyll cells and bundle sheath cells. Both are involved. Initial fixation of carbon dioxide into 4-carbon compound, catalyzed by PEP carboxylase, phosphoenol pyruvate carboxylase. Bundle sheath cells will have 4-carbon compounds transferred into it. So carbon dioxide is released for the Calvin cycle. Why C4 pathway happens? Why C4 pathway happens? C4 pathway is a mechanism of reducing water loss. Alright? Okay. Photorespiration, oxygenase activity of Rubisco. RUBP, Rubisco is supposed to use carbon dioxide and produce oxygen. If it does the reverse, it is supposed to fix carbon dioxide into higher carbon compounds. If it does the reverse, breaks down higher carbon compounds and releases carbon dioxide, that also it can do, then it is photorespiration. Respiration, what happens? We use oxygen and produce carbon dioxide, break down some organic compound, generally glucose. Photorespiration is respiration happening by the mediation of an enzyme which is supposed to do photosynthesis. So the reverse of it. Got it? Photorespiration. Rubisco binds oxygen instead of carbon dioxide, results in loss of fixed carbon and energy. Carbon being fixed would have already utilized the energy. So if you break that and it is not being used, the energy in the bond of the fixed carbon is broken down and released. Loss of energy also. Factors affecting photosynthesis. Light intensity increases the rate of photosynthesis. Rate saturates at high light intensities. Anyways, carbon dioxide concentration. Higher carbon dioxide increases rate of photosynthesis. Rate plateaus at optimal carbon dioxide levels. Water availability, indirect effect through stomatal closure. Water stress reduces carbon dioxide intake. Water stress reduces carbon dioxide intake means if there is less availability of water, that will impact the opening of the stomata. Guard cells will close, making the stomatal opening smaller or closed. So if water is not available adequately, the plant is not unnecessarily taking carbon dioxide. Generally, in a tropical country like ours, carbon dioxide is not a problem in any part of the world. We have adequate carbon dioxide available. In temperate countries and polar countries, the sunlight will be a limiting factor. In countries like ours, in tropical countries like ours, the limiting factor for photosynthesis would be water availability. So what is the law that explains the biochemical processes in terms of limiting factors? Blackman's law of limiting factors. Alright. So we have done till chapter 11, photosynthesis. Respiration in plants and growth and development in plants. 2 more chapters with plant biology. We'll do it and we'll go to human physiology. This week we will complete the syllabus map discussion of all the chapters of both 11th and 12th. And by the time I leave, I will give you the document with the syllabus map of all chapters of 11th and 12th, so that you will have enough material to use and revise. Next time when I come, you should be remembering the entire syllabus map of both 11th and 12th. Anything that is mentioned from that, you should know. Simple, something about everything. That will be level 1. Alright? Okay. Any questions, any doubts, any clarifications required? Please do ask. Otherwise, we will break for the day. 2 more sessions that you, 1 more session that you have. Do revision from your post-test analysis notes before you take the test. Alright? Only revision from post-test analysis notes and maybe the notes that syllabus map and level 1 notes that can be revised. But before exams, post-test analysis notes is what should be revised. So 2 hours from 2 to 4, you do post-test analysis notes revision. No, you are writing test now, no? 150. Okay. Anyways, all the best for your test then.